you. John Matthews, New Plymouth. There's two questions. The first one relates to the treatment stem cell therapy for secondary progressive. We've been involved in the studies in Australia, the clinical trial, phase 2 clinical trial in Australia, that is not for secondary progressive. Tish Multiple Sclerosis Research Centre in New York are planning some interesting therapeutic advances with secondary progressive stem cell activity. So could we have a comment on that, please? And the second one is we have this comment from you, and thank you, Travis Stiles, and thank you, Brett Drummond, for being here, on your Novo, I think it was 117. You didn't actually take us through whether there is any promise in that product. Thank you.
Well, I can take the first question. In terms of the trials that are going on in Australia at the moment, in both Sydney and Melbourne, I believe, I haven't seen any published data from that yet. I've actually scheduled a conversation with one of the lead researchers based at St Vincent's in Sydney to find out some more information about that. But yeah, I can't comment too much, not having seen the data. In terms of the Tish study in New York, we actually did an interview with one of their lead researchers a couple of months ago in February discussing those studies. So they have been looking at people with secondary progressive MS. The difference from the main one that I talked about with that is they're using a different type of stem cell. So they're using a type of stem cell known as mesenchymal stem cells, and these are cells that can differentiate into the type of cells that can cause repair. So that's what they're looking at. It's a completely different form to the HSCT that we talked about. They're looking at doing things that can repair the damaged myelin. They've done a small phase 1 trial that had about 20 people in it. That was really just a safety trial. They found that they didn't really have any serious adverse events from that, and they've just received FDA approval to move into a phase 2 trial for that, and that's sort of all in the planning stage at the moment. We are in discussions to have an interview with Dr. Saud Sadiq, who is the main person behind the Tish, who's been looking into these mesenchymal stem cells for around a decade now, if not longer. So we're hopeful that interview will go on shortly on MStranslate.
So I'd follow the mesenchymal stem cell thing is, again, I have no doubt that there's some promise there. My problem is what I mentioned before, it's the scientific justification. It doesn't appear that the cells themselves, the research I've seen on the basic side, it doesn't appear that the cells themselves are necessarily required for the benefit they're seeing. It seems like there's either some secretable factor. I've heard some things thrown around that people have talked about that they've isolated through different studies. And again, this goes back to the development. You know, everybody's kind of doing their own thing, and instead of trying to characterize what it actually is that's beneficial and why, until I say, okay, the cells are required, they get into the brain, they do special stuff, and you need that specific cell type at this dose, this duration, you need to isolate them in this protocol. I feel like we're skipping ahead because the guy who's been doing it for a decade is the guy running the clinical trial. If it works, is he going to run around to 1 million patients doing stem cell infusions with his stock cell line, managing it that way? The development's the important critical part to making this a widespread accessible drug or treatment. And that seems to be completely absent from a lot of these cell-based therapies that we're seeing right now. And I hope, I'm excited for any promise. When patients get better, I'm excited about that. But the problem is, can we get it beyond a small cohort? Can we get data, scientific justification, and then a proof of like a mechanism that supports the proof of concept that can be widespread, applicable? That makes sense.
That did make sense.
Novo 117.
Novo 117. So no, it's terrible. Don't show me. So I didn't show the Novo 117 data today because it's preliminary, but it's not. Novo 117 has worked fantastically well. It turns out it actually crosses the blood-brain barrier on its own, so we don't have to stab people in the eye. People are excited about that, and we also are seeing both in cell culture models and in rodent models, we're seeing good effects. So much so that we've already actually begun working on some modified derivatives that have stronger half-lives. So part of the reason I didn't get too far into Novo 117 is because we think that we can make a less frequent injectable version. So likely we're going to have another version, Novo 326, or I have no idea what we're naming it, that is going to be better. So why I emphasize the proof of concept is because I think Novo 117 works great. We're actually doing large-scale proof of concept trials at a couple different collaborator sites. The only thing is I think we can improve upon how we deliver the drug.
Thank you. Is there another question here?
Yes. Yeah, g'day. I just about the vitamin D article that you had up there. It says that the high dosage in MS patients is safe. I've had neurologists in the past tell me that prolonged high levels of vitamin D in my body is unknown. It's not necessarily known what the effects of that could be. Just a bit of clarity on that, whether you suggest that we go, Professor Jelinek's suggestion of 150, unit, trying to keep it at that level or whatever. Is that article correct in saying that you can go higher in that, or just a bit of clarity in that area would be great?
Firstly, I'm not a clinician, so I don't want to sit here and give personal information about what sort of things you should be doing treatment-wise. The study showed across the length of the study, which off the top of my head I can't remember, but I think it was 6 months, they didn't see any adverse safety effects to that. Longer term than that, I don't think, that study certainly didn't show anything from that. I think Debbie at some point might talk about prevents, and I'm not sure exactly the length of time that they're looking at for these sort of things. But part of the prevent study is also going to be looking at different dose levels to see what sort of dosage we need to see an effect, if we see an effect, and safety issues of that as well.
I will say vitamin D is a fairly easy drug to get out of your system. You'd have to dose yourself pretty high, I think, to get toxic levels. However, doing it for a year, that's probably something you don't know about that. But I do want to ask, so my understanding of you're on. No, I'm going to ask you a question. So the study you talked about the immune effects. Was that in? Because I don't remember them doing the immune panels in humans. Was that culture-ish?
That was mostly culture-ish. Yeah.
So I want to point out in that vitamin D study, when they made these huge extrapolated, it can reverse MS symptoms. They actually didn't even do they didn't take cells from patients. They said this level of vitamin D in a dish of cells affects the immune cells this way, completely independent of any other systemic thing. And then they said, if we dose humans with this level of vitamin D, those levels are reached in the blood. That was it. And it didn't kill them. That was it. And we stretched that to say we could reverse symptoms. So
all of that was in the extended MStranslate article.
Yeah.
We have another question? Let's go over here first, and then I'll go to you.
Maria Sampson, field worker from parts of the north. I've got two questions. One is, I know how scientists are very protective of their work, having been married to an academic for a long time. Are you aware of anybody chasing their tail with this information about development of your product? That's one. And secondly, realistically, how far are you from approaching companies for clinical trials?
Could you clarify chasing your tail?
Yeah, the first one, we all keep all the scientists kind of keep it all secret.
Yeah, so the nice thing about doing this from a company standpoint is I would love it if somebody else started doing some work on it, because we own the intellectual property. So if they did stuff, they can't make a drug out of it. So thanks for proving my stuff works. That's fantastic. That's why I don't feel the need to make a lot of this secret, because intellectual property, patents, things like that protect us from having other people kind of try to scoop us in that sense. In academia, it's whoever says that it's a thing first, right? So that's why people keep things more secret. And there was the second part?
How far away?
Oh, how far away? So that's a good question. If Novo 117 ends up being our lead compound and we stay with that, we're probably another 1.5 years away from doing formal IND-enabling studies. So probably looking at applying for clinical trials between 2 and 3 years. But if it turns out that our next generation of molecules work, you probably tack on another year to that.
Okay. What about, will you kind of try and get it to be multi-center?
I'm a big fan of phase 1/2A studies, where when you have an unmet need, such as remyelination in MS is something that nobody's been able to do, I like to combine your safety studies with, I like the idea of combining a safety study with a very small indicator of efficacy study. And as a result, I think it's good to try to identify a very narrow population where you think you might see your best results. Because you're not trying to say this is a huge thing. You still have to do 2 and 3, or 2B and 3, or even 3 alone if the results are strong enough. So I actually have been working diligently on trying to figure out what the best small group of centers might be and what patient cohorts might best fit that model. In addition to that, because it's an unmet need, I've also been looking at what patient populations really could benefit the most from something that might work. In New Zealand is a really interesting place to me because some of the restrictions on when there's access to care. From a very selfish and business pragmatic standpoint, the cutoff for when disease-modifying agents are kind of disallowed is a tremendous opportunity for a phase 1/2A trial because you don't have the confounding variables of standard of care because everybody's on a different cocktail of things most of the time. I'm not saying that's what I'm going to do, but that is a tremendously appealing idea that's definitely on my radar, and it's those types of opportunities that we think about when we think about where we want to do our trials.
I'm working diligently to get him to come to Australia and New Zealand. The glowworms helped.
Although Australia's trying really hard to keep me out, so
That's true.
Hi, Ray. Just another question. I'm a pharmacist. I've also stricken with MS. I just have a question about the stem cell therapy. I see you did a brilliant explanation, firstly, of the autologous HSCT. Was there a reason why you limited it just to a HSCT? And another question was about your organization, MStranslate. I just wanted to know if MS members can access that and what would they get out of it? Would it be a free membership, or do you screen articles and what do you give back to MS members?
So with the first question in terms of why I only really focused on HSCT, do you mean instead of HSCT as well, or in terms of other types of stem cell therapies?
No, you explained why not others, but I mean, when people mention that they've been for stem cell therapy, they just say, oh, I've been for HSCT. They often forget to leave out the autologous. So is there a difference?
There is a difference. I mean, if it's not autologous, that means that you're getting a bone marrow source of cells from another individual. But
that's not being done as far as I know right now.
As far as I'm aware, all of the MS ones are autologous, so you're taking out your own cells. Obviously, that's going to, if the whole principle is we're taking out cells that have been autoreactive, but then putting back in those same cells, this is where, after a period of time, and assuming that it does work, that it could come back. And this is again why I wouldn't say that it's necessarily a cure. It's essentially putting those same cells back in, but as you described it last night, it's putting them back in a baby form before they've seen whatever's made them go and start attacking. In terms of MStranslate, yes, you can all freely access MStranslate via the website, via Facebook, via Twitter.
You should have put it up on the screen right now.
It's all there. I have business cards around somewhere. If you want to grab one off me at lunch that has the website on it, which will have all of the other details. It's completely free. There's no membership. You don't even need a Facebook account to view the Facebook page. We know that one of the things that we've become acutely aware of over the time is using platforms like Facebook. While they're great to disseminate information, Facebook is a platform that uses your personal account, and we know that not everyone doesn't necessarily want to associate their personal Facebook accounts with different MS pages, so you can even access that page without having to log into anything. But it's completely free, no membership required in terms of what you will get out of it. Hopefully all of the things that we talked about in terms of, we're posting daily with research updates in easy to understand formats. There's interviews with researchers from around the world, invitations to participate in these hangouts that we do. And I guess the point that I didn't really mention is that all of the content that we create is very open to suggestion, and oftentimes we have people who follow us who say, I'm really interested in this, can you do a summary on that for me? And So, if you have something that is of particular interest or something that you don't understand, then, email those through or drop them on the Facebook page or anything like that, and then we're happy to create that content specifically for that. Whenever we do interviews with researchers, we always invite people from the community to submit their questions as well, which we then ask.
That's great, thank you.
Another question? Somebody else? Okay, I'm going to go. I'll go here and I'll go there. We got enough time?
Mine's more of an idea of question. I'm a neurological physio, and I think what we do know from research is that neuroplasticity and remyelination is actually really driven by exercise, and I think that's probably an idea that a lot of the population in this room aren't aware of. And one of my ideas to you guys is to get a really successful clinical trial is if you have a drug that's promoting remyelination and you also combine it with intensive exercise and rehab afterwards, I think you'll hopefully get the results that you want much, much stronger.
So that's an excellent point. And I mentioned earlier some of the connection between spinal cord injury and MS in the regenerative potential. Spinal cord injury is really easy because you cut a spinal cord, all motor function beneath the injury is gone. And so exercise and functional stimulation of those pathways is easy to do because what's not moving, let's try to get it to move again. But what she just mentioned is extremely true. The pathways that are damaged heal so much better if they're being coaxed into activity. And exercise is a huge component of that. And I would add to that activity specific to the function that's been affected is extra important. Being active, getting vasodilation and getting your heart rate up, all those things are extremely beneficial. But rehabilitation and stressing of the systems that have been affected and combining with the therapy, that's always going to be a huge part of what improves outcomes. And you're 1,000% right. It's easy to incorporate that into spinal cord injury because physical therapy is a huge component of that already. The difficult part with MS is lesion location might change the need for, or the types of modalities that might be needed. As those become more standardized, absolutely, I would advocate for incorporating those into any trial.
Thank you. Over here.
Hi, I'm Tina, and my question is regard to the design of Novoron Bioscience. I'm just wondering, in future trials, are you planning to use that drug just on its own or with an immunosuppressant? Because I don't see the point of building up myelin just to have it crushed by a breaking immune system.
So if a patient's on a disease-modifying agent, we obviously wouldn't take them off. But we wouldn't add anything to it either. The truth is, if you do too much if we try to add additional immunosuppression, it kind of will affect the dynamics of the trying to say this in a not too sciencey way. We would leave whatever other current therapies were intact. The difference between what we would be doing is we're in acute therapy. If you have an attack, we would treat you for a specific period of time, which those trials would need to be ongoing to assess how long those treatments would be to take place. It would not be long-term, lifelong therapy. It would be lesion-specific, or in the case of chronic lesions, we would have to understand how those dynamics work as well. But if your lesions are under control, there's not really a need to have this foreign entity in your system. So it only would be to treat the lesions that exist acutely, not for chronic treatment. Does that answer your question?
It's just regarding vitamin D. It's something I've been taking for a number of years now, and it's been enormously beneficial for me. And I can't be without it. And interestingly, in regards to the media, yes, a few years ago they were saying that vitamin D in tablet form has no beneficial effect of any kind, whatever. So I stopped it for a few months, and I found that actually I became a lot weaker. My walking became a lot more unsteady. I was, I was in a terrible state, so I went back on it, and I came right again. It took me a while, but I have to say, according to people that I've read and everything, the safe upper limit for vitamin D is actually 10,000 international units a day, and that's fine to be taking it. They did a study in Canada.
I think you're talking about ingesting, or
just taking it in tablet form.
Yeah, I mean, I think that's well, I mean, I think that's well within the range of, I mean, I think you can go higher than that as well. I don't want to speak, there was, no?
That one was 10,000. That one was 10,000 across the
But,
time frame.
but yeah, vitamin D is not, where's the clinician? We need somebody to answer the question.
According to Dr. Dominic, who was a clinician,
Yeah.
but also I know they did a study in Canada where they were giving people on trial 40,000 every day.
Yeah.
And for a period of a year, and it had no sort of side effects or problems or whatever
Yeah.
after that.
Doesn't surprise me.
Yeah. So, sorry, one question as well. I know that Dr. Yamanaka won the Nobel Prize in 2012.
The stem cells guy?
Yes, but he's sort of focusing on stem cell surgery to do with spinal cord injury.
He's the guy who invented inducible stem cells. So
That's
take
right.
it.
That's
So
right.
take
I
it.
mean, would that have he done work on
So
this
here's
as well?
here's what I'm saying. Could it be useful? Yes. Is it anywhere near for anyone in this room to be concerned about? No. There is so much that needs to be done on genetically modified cells that would be inducible pluripotent stem cells before it can be a ready therapy. And I want to be very clear on this because there are companies in several places in the world who prey upon patients who are wanting to find hope with elegant technologies, with fancy potential future capabilities that have no real scientific linear justification towards the actual disease. Yamanaka's work, the reason that he so recently received that Nobel Prize is because embryonic stem cells are stigmatized for numerous reasons. And so he was able to reprogram your own stem cells, which then minimizes immune problems. You can reprogram them to be a whole bunch of different progenitor cell types that can be nerves or liver cells or cardiac cells. But we are just scratching the surface of how to handle that technology, how to apply it to disease. I've actually worked into some of this in how to differentiate cells into specific things, and it's fascinating, but we are very young on the scale.
Well, and that's why I think, and this is again something I'm sure said when we were talking about it, why finding good places to get information from is so important. When we were sitting around yesterday working through some of this stuff, you type stem cells into Google, stem cells or stem cells MS, I can't remember exactly what our search term was, but 2 of the top 3 hits had just incredibly terrible information.
2 of the top 3.
You know, I think one of them was here. One of them was saying something along the lines of, from turning up, meeting the doctor that would be treating you, having your stem cells harvested, having the treatment done, could all be done in 3 hours. Now,
anybody who thinks that's possible? Anybody? No.
and that was one of the top hits. So it's just, you just need to be really careful about what